Have you ever really thought about how a new medicine gets made? I mean, it's not some single eureka moment in a lab. It is a grueling, high-stakes gauntlet. It's this incredible journey that starts with literally millions of possibilities, and if you're lucky, ends with just one. Today, we're diving into that journey.
So let's just start with the big question right up front. Why is this so hard? I mean, we hear about medical breakthroughs, but the reality is, for every single success story, there are millions upon millions of failures. What makes finding that one effective compound so unbelievably difficult? Well, the answer isn't just dumb luck. It's all about having a highly structured, meticulously planned strategy. It's a scientific game plan designed to systematically weed out all the failures and find that one potential winner. It's how you find a needle in a haystack without going insane. And that game plan? It's got a name: the screening cascade. You can think of it as the central operating system that organizes this entire massive effort of drug discovery. So what exactly is a screening cascade? Just think of it like the rulebook for the entire project. It's a flowchart of experiments that tells this huge team of scientists, chemists, biologists, you name it, exactly what to test and when. It keeps everyone on the same page, which is absolutely critical when the stakes are this high. Now, this cascade has 4 really crucial jobs. First, it lays out which experiments to run. Second, and this is just as important, it determines the most efficient order for those tests. Then it defines the clear, hard cutoff for what counts as success at each stage. But its most important job, it acts as this relentless filter, making sure only the absolute best, most promising compounds survive to the next round. And that brings us to the real heart of the process, the gauntlet itself. And listen, this isn't just one test. It's a whole series of increasingly difficult challenges, each one designed to push every single compound to its absolute limit. Let's walk through the first few of these filters. The first, and honestly, the most brutal filter, is potency. This is where the vast majority of compounds get eliminated because it answers the most basic, non-negotiable question a scientist has to ask. Does this work at all? Potency basically asks, does this compound even do anything? Does it actually interact with and stop the disease target, whether that's a specific protein or the entire bug? If the answer is no, it's game over right then and there. Without potency, you don't have a drug candidate. You just have a random chemical in a vial. So how do scientists measure this? They set a really specific cutoff. For example, in a malaria project, a compound might need to show potency at 500 nanomolar or less. And here's the key. With potency, a lower number is always better. It means you need less of the drug to get the job done, which makes it more efficient and, hopefully, safer down the line. But look, being potent is just step one. A compound can be incredibly powerful in a test tube, but if it can't actually work inside a human body, it's completely useless. And that brings us to the next big filter: solubility. This is basically a test of how well the compound dissolves in water. And since our bodies are, mostly water, this is absolutely critical. For a pill to work, the drug has to dissolve so it can be absorbed into your bloodstream. Not only that, but if a compound doesn't dissolve well in the lab, it can give you all sorts of messy, unreliable data in other tests. So poor solubility is a quick way to get kicked out of the running. Okay, so let's say our compound is potent, and it's soluble. Great. Now for what is arguably the most important test of all: toxicity. Is it safe? This is where things get really tricky, because it's a very delicate balancing act. This right here is the fundamental conflict of pretty much all of drug discovery. You need a compound that is deadly to the disease. That's goal number 1. But at the exact same time, it has to be as harmless as possible to our own healthy cells. That's goal number 2. The perfect drug is a selective killer. Scientists actually measure this balance using something called the selectivity index, or SI. It's a pretty simple ratio. You take the dose that harms our cells and divide it by the dose that kills the pathogen. The bigger that number, the wider the safety margin, and the safer the drug is. A high SI is what every single project is chasing. But here's where it gets really fascinating. Safe enough isn't some universal standard. For a disease like malaria, the safety bar is set incredibly high. You need an SI of 100 or even more. But for a life-threatening cancer, scientists and patients might be willing to accept a much riskier compound, maybe one with an SI of just 10. The entire risk-reward calculation changes depending on the disease you're fighting. So we have all these tests: potency, solubility, toxicity, and trust me, there are many more. But how are they all arranged? Who gets to decide the rules of the game? This brings us to the strategy behind designing the cascade itself, this perfect funnel. Right. Who sets those cutoff values we were just talking about, and who determines the order of all the tests? It's not arbitrary at all. It all flows from one single master document. It's called the Target Product Profile, or TPP. You can think of this as the ultimate blueprint for the entire project. Before a single experiment is ever run, the team defines exactly what the final drug needs to look like. How will patients take it? A pill? An injection? How safe does it need to be? How much can it cost? The TPP answers all of these questions from the very beginning. And here is the crucial point. The TPP, the what you want to create, directly determines the entire structure of the screening cascade, the how you're going to find it. Every cutoff, every single test is specifically chosen to find a compound that meets the demands laid out in that initial master plan. Okay, so let's imagine a compound makes it all the way through. It passes the potency test, the solubility test, the toxicity test, and all the other filters in the cascade. It's a winner, right? We found our drug? Well, not so fast. This single sentence is one of the most important takeaways from this whole process. After all of that work, after surviving this incredible gauntlet, a compound that passes the screening cascade is still not a drug. And why is that? Because a whole new, even tougher gauntlet is waiting. The compound has to go through far more extensive safety testing, then come preclinical studies in animal models, just to see if it even works in a living, breathing system. And if, and it's a big if, it survives that, it faces the biggest hurdle of all: multiple, years-long phases of human clinical trials. So, the screening cascade is an absolutely essential tool. It's how you find the most promising candidates in a sea of millions. But really, it's just the qualifying round. It's the first gauntlet in a long, expensive, and incredibly difficult journey toward creating a new medicine. Which leaves us with this one final thought. After starting with millions of compounds and running them through this gauntlet, and then all the other stages that follow, only a tiny, tiny fraction ever make it to the pharmacy shelf. It really makes you wonder, what amazing combination of science, strategy, and maybe just a little bit of luck makes that one surviving compound so